The presented protocol is useful for rapidly screening and analyzing bioactive candidates in natural products to control oral biofilms. It can also be adapted for applications in other biofilm research fields. This multi-target screening and analysis method make it possible to screen multiple active components simultaneously.
Dental caries is a biofilm diet-derived, highly prevalent chronic disease. This protocol can help to identify natural products to control biofilms and, consequently, dental caries.
Begin by preparing the extraction solvent with a hydroalcoholic mixture. Use sample concentrations between 50 and 100 milligrams per milliliter of the extraction solvent and perform 15-minute ultrasound-assisted extractions in microtubes. Repeat the extraction 3 times. After each extraction step, centrifuge the sample to decant the solid residue and remove the supernatant. Combine the supernatants from each extraction step and filter them. Save the aliquots for chemical analysis and bioassays. For fractionation, dilute the crude extract sample to obtain a 100 mg per milliliter solution using the initial extraction solvent. Then transfer 1 milliliter of this sample to a preconditioned solid phase extraction cartridge with 1 gram of adsorbent and a capacity of 6 milliliters.